It's a virus! What the? The antibodies aren't working! Could this be type A influenza?
Type A influenza. This virus features numerous internal mutations and is prone to triggering epidemics on a global scale.
Hi, my name is H1N1, the common subtype of avian origin of influenza A. For both human H1N1 Spanish flu pandemic in 1918 and swine flu pandemic in 2009, I'm a member of the family of Orthomyxoviridae and genus Alpha influenza virus. I'm a negative-sense single-strand RNA virus. I have two proteins on my cell membrane. Scientists identify me as H1N1 from the subtype hemagglutinin 1 and neuraminidase 1. I cause 36,000 deaths in the United States each year. I can undergo a small variation called antigenic drift. That's why flu vaccines need to be updated every year. Another change is so dramatic that the subtype H and N change that human antibody cannot recognize, called antigenic shift. This is the time epidemic or pandemic can occur. It was the first time in 1918 pigs were infected by both avian and human H1N1 virus. At the same time, I jumped species and adapted myself to rapidly, readily, and efficiently spread from human to human and killed 50 to 100 million people worldwide. I also infected cats and dogs in 2009. I enter your body through your nose, throat, and lungs. My H proteins bind onto the salicylic acid of your epithelial cells that line your respiratory tract. I enter the cell by receptor-mediated endocytosis. I move towards the nucleus along with the enzymes and genetic materials. The cell reverse transcriptase converts me into a length of DNA, which inserts into the cell's genome to print copies of myself above of your membranes with the help of N-spike after killing your cells.
The influenza virus is said to multiply by 100 after 8 hours inside the body and by 1 million after a single day.
One very unusual inflammatory response of 1918 pandemic is a W-shaped mortality pattern where the H1N1 virus infected the group between 20 to 40 the most. My incubation period is 18 to 72 hours. You start to experience symptoms like fever, dry cough, headache, muscle or joint pain, sore throat, chills, fatigue, and runny nose. Some severe forms characterized by quick deterioration about 3 to 5 days after the first symptoms onset. Some patients will progress into pneumonia, respiratory failure, and multi-organ failure. Lab diagnosis is done using RT-PCR from a nasopharyngeal swab or washing, or samples taken from endotracheal aspirate or bronchoalveolar lavage fluid or throat swabs. For symptom relief, most people only require resting, plenty of fluids, and oral NSAIDs or acetaminophen. For severe symptoms, antiviral drugs like oseltamivir or zanamivir is needed to reduce the risk of complications. For preventative measures, hand washing, proper personal protective equipment like masks and gloves to avoid transmissions. Vaccination is very important to avoid influenza outbreaks. Scientists reconstruct the 1918 influenza virus using the preserved tissue cells to better understand how did the virus adapt within that person and became a human virus, to develop drugs that target or limit aspects of the inflammatory response. More researches are done to find out possible variants and how to predict the next pandemic.